The overall goal of this procedure is to investigate calcium remodeling in the aging brain using fluorescence and bioluminescence imaging of cytosolic and mitochondrial calcium in long-term cultures of rat hippocampal neurons, which is considered as a model of neuronal aging.
This method can help to understand key questions in the aging field, such as how remodeling of intracellular calcium contributes to excitotoxicity and neuron damage, both related to calcium overload.
The main advantage of this technique is that it is possible to follow closely calcium remodeling in individual neurons using cultures at different days in vitro from the same specimen.
The implication of this technique extends toward therapy of stroke or Alzheimer's disease, because it might reveal novel targets related to calcium overload in these aging-related diseases.
After watching this video, you should have a good understanding of how to perform fluorescence and bioluminescence imaging of cytosolic and mitochondrial calcium in short-term and long-term cultures of rat hippocampal neurons.
To begin this procedure, distribute the cover slips on a strip of parafilm in a petri dish. Then, apply 200 microliters of poly-D-lysine at 1 milligram per milliliter on each cover slip and treat overnight. The next day, wash the cover slips with double distilled sterile water every 15 minutes for 90 minutes under sterile conditions. Place the cover slips in the multi-dishes and maintain them in a humidified